So welcome everybody to this distinguished IAS lecture series. So I want to make a quick announcement because this lecture is sponsored by IAS as distinguished lectures. So it will be videotaped and will be used for undergraduate education purpose. So I feel you don't mind. But only this lecture, the remaining lecture, well, you're free to say whatever you want. Okay. So it's my real pleasure to introduce our distinguished speaker today, Dr. Juan Bonifacino. So Dr. Bonifacino grew up in Argentina, and after he received his PhD from Argentina, he moved to NIH, trained as a postdoc with Dr. Richard Clausers. He then stayed on and rose through the rank in NIH. He's now the Associate Scientific Director of CBMB of NICHD at NIH. So over his scientific career, Dr. Bonifacino has published over 100 papers that focus on the study of signaling and adaptive protein from endosome to lysosome and lysosome to TGN. And actually myself was actually got to know him when I was looking at his phenomenal study on the regimers that control the trafficking of vesicle from the lysosome back to the TGN. So he also received numerous awards and served in multiple editorial boards of important journals. And he also was invited to give distinguished lecture in many other places. So what actually struck me more is not how many awards or editorship he has served. It's actually how he treats the junior faculties. So I have myself included, and also I talked to a number of my colleagues. We all have the feeling that Dr. Bonifacino is quite generous and supportive to the junior scientists, and he has sincerely, he has tried to do all his best and sincerely help those junior scientists. In fact, this is also evidence that he had trained over 70 scientists from his lab, and the majority of them has become professors in academic institutions. So I think that speaks more about his academic achievement. So without further ado, let's welcome Dr. Bonifacino.
Good morning. Thank you very much, June, for your very kind and generous introduction, and also for inviting me to give this lecture. I'm really pleased and delighted and honored to be here in Hong Kong meeting many old friends. So as Jun mentioned in his talk, my lab has been interested for many years in the molecular mechanisms by which proteins go to different compartments within the cell, endosomes, lysosomes, so-called lysosome-related organelles. And in the course of those studies, we identified many sorting signals, adapter proteins, code proteins, and others that recognize those signals and then deliver the proteins to their correct location within the cell. But instead of presenting an overview of all that work that we have done over the years, today I've decided to present a small story of discovery, recent work from my lab that has to do with the mechanisms and functions of lysosome positioning. As you know very well in this audience, lysosomes are membrane-bound organelles that you can see in this immunoelectron micrograph of a pituitary cell stained with an antibody to a resident protein of lysosomes, LAMP1, which are mainly responsible for intracellular degradation of biomacromolecules. And they were discovered in 1955 by Christian de Duve. And lysosomes, this degradative function of lysosomes is carried out by over 60 acid hydrolases, enzymes such as proteases, glycosidases, lipases, nucleases, and others that collectively are capable of degrading virtually any biomacromolecule. And because of this role in intracellular degradation, lysosomes have been referred to as the cell's stomach and waste bags, trash processors, garbage disposal system, and other equally unattractive terms. And so because of this perception of lysosomes as trashy organelles, interest in lysosomes has remained limited to a certain area of cell biology. But this perception is beginning to change with the realization that in addition to participating in degradation of molecules coming from endocytosis, phagocytosis, biosynthetic sorting, lysosomes also underlie, defects in lysosomes underlie many diseases, including not only relatively rare lysosomal storage disorders, but also more common neurodegenerative diseases such as Alzheimer's disease, Parkinson's, Huntington's, and ALS. So there is a lot more interest in understanding the biology of lysosomes for this reason. In addition, over the past 10 or 20 years, it's been found that lysosomes also participate in many other cellular functions, some of them degradative and others non-degradative, including autophagy, which is a whole field by itself, antigen presentation, killing of intracellular pathogens, lipid metabolism, detoxification, cell adhesion and cell migration, tumor invasion and metastasis, apoptosis, metabolic signaling, and even gene regulation. So really lysosomes have now reclaimed center stage among all the cytoplasmic organelles because they participate in many critical cellular processes. And in this talk, I'm going to tell you about an emerging property of lysosomes, which is their positioning and motility within the cell, and in particular the mechanisms of lysosome positioning and motility and the physiological functions of that property, those properties. So what do I mean by lysosome positioning? So this is a HeLa cell expressing LAMP1, which is a lysosomal membrane protein tagged with GFP. And as you can see, lysosomes are found throughout the cytoplasm. They are pretty much everywhere in the cytoplasm. There is a juxtanuclear cluster, but there are also many peripheral lysosomes that even reach into the protrusions of the cell. So they are everywhere. So that's their position within the cell. And motility, if we now perform live cell imaging of this cell, you can see that while some lysosomes are stable, either at the center or the periphery, others move back and forth between these two populations along microtubule tracks. And this has been known already for quite some time. Outward movement towards the plus end of microtubules in the periphery is mediated by microtubule motors known as kinesins, whereas inward movement is mediated by a different type of microtubule motor known as dynin. So lysosomes couple to kinesin and dynins, and then that allows them to move bidirectionally between the center of the cell and the periphery of the cell. So in the first part of my talk, I'm going to tell you about recent work that provided insights into the mechanisms by which lysosomes couple to kinesin for transport to the periphery of the cell. And in the second part of the talk, I'm going to tell you about what the physiological importance of that outward movement is. Now, this is a project that we didn't set out to study. We just stumbled upon this problem in the course of studies on another problem, which is a genetic disease that we have been studying for many years in the lab that is known as the Hermanski-Pudlak syndrome. This disease is an autosomal recessive disorder characterized by hypopigmentation, bleeding, lung fibrosis, and colitis. what's common to all of these organelles is that, to all of these organs, is that they contain so-called lysosome-related organelles, which are defective in this disease. For instance, the hypopigmentation is due to defects in melanosomes, while the bleeding is due to defects in platelet-dense bodies, which is another type of lysosome-related organelles. The lung fibrosis is due to defects in lung lamellar bodies, and the colitis has an inflammatory origin. And these two symptoms, the lung fibrosis and colitis, are the ones that lead to death in many cases, starting on the fourth decade of life. So many years ago, we found that one cause of Hermanski-Pudlak syndrome is mutations in the beta-3A subunit of the heterotetrameric adaptor protein complex AP3. So mutations in AP3 are one cause of Hermanski-Pudlak syndrome. However, genetic studies have shown that this disease is caused by mutations in many different genes, up to 9 so far in humans and more than 15 in mice. And unlike AP3, which is a known subunit of an adaptor protein complex involved in protein trafficking, the genes that are mutated in the other forms of Hermanski-Pudlak syndrome encode proteins of no known function and no homology to any other proteins in the human genome or other genomes. And for that reason, it has been difficult to elucidate the function of those other proteins. And work done in my lab, also in the lab of Esteban de la Angelica, a former postdoctoral fellow from my lab, and in other labs, have shown that those other genes that are mutated in Hermanski-Pudlak syndrome encode subunits of 3 other complexes that we named block 1, block 2, and block 3. The most complex of all is this block 1 complex, which is composed of 8 different subunits. And it took us a while to actually figure out that they were all part of the same complex. Now, interest in block 1 in particular has risen with the finding that some of the subunits of block 1, in particular this binding, is a schizophrenia susceptibility protein. It's encoded by a schizophrenia susceptibility gene. And because schizophrenia is much more common than Hermanski-Pudlak syndrome, you will find block 1 mentioned in relation to schizophrenia more often these days. Okay, so now the AP3 complex is an adaptor protein complex, which is a component of a protein coat associated with endosomal tubules, and which is involved in the sorting of transmembrane proteins to lysosome-related organelles such as melanosomes. An example of those cargoes is the enzyme tyrosinase, which is a key enzyme in the biosynthesis of the pigment melanin. So if there are defects in the AP3 complex, tyrosinase is not sorted to melanosomes, and then there is a pigmentation defect, which explains the hypopigmentation in these patients. Block 1, Block 2, and Block 3 are believed to function in the same pathway of transport from endosomes to lysosome-related organelles, but their biochemical functions are less well understood, at least in mammals. There is work from Christian Ungermann that you will be hearing from today that has shown that block 1 is actually a regulator of Rab GTPases and in yeast, and it's possible that it may play a similar role in mammalian cells as well. Now, we've been studying the molecular functions of these block complexes for quite some time with very little success. This is probably the most difficult project that I've worked on. And that's due in part to the fact that these complexes are not very abundant in cells. And it has 8 subunits. So if you express an epitope tag or GFP tag subunit in cells, you'll see a lot of cytosolic staining because you are overexpressing the transfected protein. And immunofluorescence microscopy for the endogenous complex in general doesn't work because the complex is not abundant at all. So that has really made it difficult to elucidate the functions of these complexes. So one approach that we took to get at the function of this complex was to tag one of the subunits of block 1, BLOS2, with an affinity purification tag to express that protein stably in mammalian cells and then use it to purify the complex and associated proteins that were identified by mass spectrometry. And what I am going to tell you about now doesn't really have to do with directly with the block 1 complex, but we found something unexpected. First of all, in this affinity purification and mass spec, we identified BLOS2, which was the bait used in this analysis. These are two independent purifications, and this is the peptide number that we obtained in the mass spec. In addition, we found the other 7 subunits of the block 1 complex. So the approach worked because we could easily identify the bait and also the other 7 associated proteins that were already known. But curiously, we obtained 5 other previously uncharacterized proteins, the peptide numbers of which were comparable to those of the known block 1 subunits, and in some cases they were even higher. So these were not minor contaminants, they were as abundant as the known subunits of the block 1 complex. So we were puzzled, and we thought, wow, so 8 subunits are not enough. This complex has even more, perhaps 5 more subunits. So then what we did was to tag 2 of these subunits, the most abundant ones, LOH12CR1 and C17OR59, which were previously uncharacterized open reading frames, and then repeat the affinity purification and mass spec analysis for those 2 proteins. And when we did that, and we did it in many different ways, multiple times with the tag at the N terminus, at the C terminus, with similar results in all cases, we found that these 2 proteins co-purified not only with themselves, but also with a subset of the block 1 subunits, which led us to hypothesize that in addition to the block 1 complex, which contained these 8 subunits, there was a related complex which contained another 8 subunits, and which we named, at least provisionally, BORC, for block 1 related complex. So this was a hypothesis, and we performed many biochemical analyses to demonstrate that indeed these 8 proteins were part of a multi-protein complex, and that this multi-protein complex was distinct from block 1. And I'm not going to show you all that biochemical evidence. I'm just going to show you one piece of evidence. So the hypothesis then is that there was a block 1 complex with 8 subunits that was already known, and that we had found a related complex which shared 3 subunits with block 1, BLOS1, BLOS2, and SNAPin, but in addition had 5 previously uncharacterized proteins, 3 of which were renamed by us, myrlysine, lyspersin, and diaskedin, all of which refer to, as you are going to see, the localization of this complex to lysosomes and their role in lysosome positioning. Okay, so the piece of evidence that I found most convincing for the existence of this complex was to reconstitute the whole 8 subunit complex in E. coli by expression of the 8 subunits from a polycistronic expression vector. And 2 of the subunits were tagged, one with a 6-his tag and the other with GST, and those tags were used to perform a double purification of this complex. We then cleave the tags, we run this complex on a gel filtration column, and this is what we saw. All of these bands sort of eluded together on this super 6 column, and the identity of the bands was confirmed by mass spec. Lyspersin migrated as 3 bands because it was proteolytically cleaved during the purification, and all the other proteins were here. So all of them eluting together. So indeed, this is part of a new complex that we named BORC. Okay, so we started out wanting to understand the function of block 1, but instead of elucidating the function of block 1, we found a related complex about which we knew even less. We knew nothing about this complex. So we decided then to begin to investigate the function of this complex. And the first thing that we did was to epitope tag one of the subunits, myrlysine, with various tags. And I'm going to show you results with GFP. We then transfected cells with that GFP tagged myrlysine construct. And this is what we saw. We found that myrlysine localized to these vesicular structures in the cytoplasm of the cell. This is the nucleus, and this is the cytoplasm. So myrlysine was associated with these vacuoles that colocalize with lysotracker and with other markers for lysosomes. So myrlysine was associating with lysosomes. And you can see here a higher magnification of this image here. You can see that in green, myrlysine associates with the limiting membrane of the lysosome, whereas lysotracker labels the inside of the lysosome. So it was associated with the limiting membrane of the lysosome. By immunoelectron microscopy, we also saw that myrlysine in the large gold particles was associating with these vacuoles that also contained LAMP1 labeled with small gold particles, which you may not be able to see from the back, but believe me, they are there, and they are labeled with these arrows. So clearly, myrlysine was associating with lysosomes. And there is also some evidence from proteomics analysis of lysosomes that the subunits of the BORC complex were already there, except that they had not been identified because their identity and function was not known at the time. Okay, so myrlysine associates with lysosomes, and what does it do there? In order to address that question, we then knocked out the gene encoding myrlysine in HeLa cells using CRISPR-Cas9, and this is endogenous myrlysine in wild-type HeLa cells, and this is in the myrlysine knockout cells using two different antibodies to the protein. So we were able to obtain this knockout cell line. Interestingly, the knockout cell line was viable, was perfectly viable, so this is not an essential gene. And in addition, the knockout cells also grew at the same rate as the wild-type cells, so it didn't have any effect on the ability of these cells to grow. So, okay, so did they have any problems? So we then looked at lysosomes, obviously, because that's where myrlysine was found. And in wild-type cells, you can you see from the back very well this, or should we dim the lights a little bit? What do you prefer? Oh, okay, you can probably see it. Okay, this is the nucleus of the cell, and these are the lysosomes in the cytoplasm, as I showed you at the beginning of the talk, stained with an antibody to LAMP1, CD63, and LAMPTOR4, which is a subunit of the regulator complex, which is also associated with lysosome. So this is the usual dispersed distribution of lysosomes that you see. But we then looked at the localization of these markers in the myrlysine knockout cells, and we were actually very pleasantly surprised that the lysosomes were clustered in the juxtanuclear area, and they were depleted from the periphery of the cell, which immediately told us that myrlysine was involved in regulating lysosome positioning. In contrast to lysosomes, other organelles, such as early endosomes, labeled for the transferrin receptor, mitochondria labeled with mitotracker, and the ER labeled with ER tracker had a normal localization in the myrlysine knockout cells as compared to the wild-type cells. We couldn't see any changes in the distribution of even something that sits in the same pathway as late endosomes and lysosomes, early endosomes. So myrlysine was specifically involved in regulating the positioning of lysosomes. And I must add late endosomes, because in most cases we are not able to distinguish one organelle from the other. We then perform live cell imaging, and this is the picture that you've already seen at the beginning of my talk, with many lysosomes moving back and forth between the center and the periphery of the cell. And this was the imaging of lysosomes in the myrlysine knockout cells. The lysosomes sort of stayed in the central area of the cell, and they just jiggled in place. They vibrated in place, but they were unable to move to the periphery of the cell. So these cells had an inability to translocate lysosomes from the center to the periphery of the cell, a process that we know is dependent on kinesin. This role of myrlysine was not limited to HeLa cells, but we obtained similar results in other cell types. And I would like to show you the results that we obtained in rat hippocampal neurons in primary culture. This neuron has been transfected with a construct encoding LAMP1 yellow fluorescent protein in this case. This is the soma of the neuron. These are the dendrites, and this is the axon, labeled with a marker for the axon initial segment here. And as you can see, lysosomes are found everywhere in the neuron. They are found in the soma, in the dendrites, and also in the axon. This is unlike other organelles. For instance, transferrin receptor containing endosomes have a polarized distribution, and they are found in the soma and the dendrites, but not in the axon. So unlike early endosomes, lysosomes are non-polarized in these cells. So they move back and forth between the ends of the axons and the dendrites and the soma. So we then imaged the LAMP1 and myrlysine GFP in neurons transfected with these constructs only in the axon. And what you may be able to see here is that, so you will see the lysosomes moving back and forth, and you will see occasionally some lysosomes that contain associated myrlysine as well. So myrlysine is also found in association with these lysosomes that move from the soma toward the distal axon. This can be analyzed by performing this kind of chymograph analysis, and these lines that have a negative slope correspond to the anterograde transport that move towards the distal axon. And you will see that some of them contain both LAMP1 and myrlysine, as you can see here. There are many more LAMP1 containing structures, and some of them have faintly associated myrlysine, but there are many others that are retrograde carriers, and those do not contain myrlysine for the most part. So myrlysine is mainly associated with lysosomes that move toward the distal axon. And in the case of neurons, we performed RNAi by transfecting the cells with an shRNA to lyspersin, another subunit of the Borg complex. And we found that whereas LAMP1 is non-polarized in the control neurons, it's in the dendrites and it's also in the axon. In the lyspersin depleted cells, there is a lot less staining for LAMP1 in the axon, and also a somewhat reduced staining for LAMP1 in the dendrites as well. So also in these neurons, the Borg complex appears to be involved in moving lysosomes to the periphery. Now, as I already alluded to, the movement of lysosomes to the periphery is mediated by association with the kinesin motor. And work by different groups have shown that lysosomes are coupled to kinesin 1 by virtue of a complex that involves a small GTPase, RL8, which is a member of the ARF family of small GTPases, which recruits an effector known as skip by virtue of an interaction with a RAN domain. And this effector, in turn, interacts with the kinesin light chain, which is part of a complex with the kinesin heavy chain, or KIF5. And so this is how lysosomes were previously shown to couple to to kinesin. So we wonder, where does Borg fit in this picture? Is it involved in any way in this chain of interactions that couple lysosomes to kinesin? So we decided to look at the localization of some components of this complex. And we first looked at the localization of RL8. And so these are HeLa cells stained for endogenous LAMP1. And can we dim a little bit the lights a little bit more? I think it's more striking if you can. Okay, now it will be better. So it's better that you see the pictures and not me. So, okay, so these are HeLa cells stained for endogenous LAMP1. And this is the transfected RL8 isoform B, RL8B, stained with GFB. And you may be able to see that there is a very good colocalization of RL8B with LAMP1. So in other words, RL8 associates with the lysosomal membrane, as previously shown by others, including Sean Monroe and others. And we then looked at the localization of RL8 in myrlysine knockout cells, and this is what we saw. The lysosomes were clustered, as I showed you before, but RL8 was now much more cytosolic. It was not associated with the lysosomal membrane, indicating that indeed myrlysine was required to recruit RL8 to the lysosomal membrane, initiating the chain of interactions that led to coupling to kinesin. We also, so far I've shown you most of the results for the knockout or knockdown of myrlysine, but we also did similar experiments for the other subunits of the Borg complex, knockouts or knockdowns, all of them with similar results. And I'm just going to show you some examples of that. When we knock down using RNAi the other subunits of the Borg complex, we also saw that lysosomes were clustered and that RL8 was more cytosolic. In some cases, the phenotypes were partial, perhaps because the RNAi was not 100% efficient, but as compared to the untreated cells, you can see that RL8 is a lot more cytosolic. So the knockouts or knockdowns of all the subunits of Borg give essentially the same phenotypes concerning lysosome clustering and dissociation of RL8 from the lysosome. We also tested the effect of knocking down or out the 5 subunits that are specific of the related block 1 complex. And when we did that, we observed that the lysosomes had a normal distribution and RL8 was perfectly associated with lysosomes. So although block 1 is related to Borg, it appears to be involved in a different process. So on the basis of these experiments, we concluded then that Borg regulates lysosome positioning and motility through recruitment of the RL8 GTPase to the lysosomal membrane. And block 1 doesn't do that. So what does block 1 do in mammalian cells? We still don't know very well, for sure, but with the knowledge of what the molecular function of Borg is, we are going to go back and test some other hypotheses as to what the molecular function of block 1 might be in this system. Now, work from several labs, including the lab of Grasa Raposo, suggests that block 1 is associated with tubular endosomes and in that it may be involved in extending these tubular endosomes, perhaps for the purpose of transporting proteins from endosomes to the lysosome-related organelles such as melanosomes. Okay, so in summary, Borg is associated with the lysosomal membrane, in part due to a myristoylation site at the amino terminus of the subunit named myr-lysine. That's why we named it myr-lysine, myristoylated lysosomal protein. But that is probably not the only subunit that mediates association with the lysosomal membrane. Then Borg functions to recruit RL8 to the lysosomal membrane. We hypothesize that Borg might be a GIF, a guanine nucleotide exchange factor for RL8, because that would be the phenotype of a GIF knockout, that RL8 would dissociate from membranes. But today we have not succeeded in demonstrating GIF activity of Borg for RL8 in vitro assays, but we're still trying. That remains still a very likely explanation. And then RL8 recruits the rest of the machinery for the movement of lysosomes toward the placenta of microtubules in the cell periphery. Okay, so now I'm going to move on to the second part of my talk, which is to address what is the physiological importance of moving lysosomes to the periphery. Does it matter at all whether the lysosomes are everywhere in the cytoplasm or at the center of the cell? Because as I said before, the knockout cells are perfectly viable and they grow at similar rates as the wild-type cells. So does it really matter? Well, not for viability or replication of the cells. However, as I mentioned at the beginning of my talk, lysosomes have many functions, and so the question was whether positioning and motility were involved in regulating at least some of these functions. And I'm going to show you some of the results that we obtained in this line of work. First of all, we noticed that whereas wild-type HeLa cells have some relatively faint staining for LC3, which is a marker for autophagosomes within these cells. So wild-type HeLa cells have a basal level of autophagy going on at all times. The myr-lysine knockout cells have elevated levels of LC3 staining, as you can see here. We also analyzed the myr-lysine knockout cells and knockout cells for some of the other subunits of the Borg complex by immunoblotting. And you may be able to see this is wild-type cells, and these are the myr-lysine knockout cells analyzed for LC3 levels, that the levels of LC3 are elevated. And rescue with the myr-lysine cDNA causes the LC3 levels to go down again. So also using immunoblotting, we can see that the knockout of myr-lysine increases the levels of LC3. And the same is true for knockouts in some of the other subunits of the Borg complex. Now, as I said, LC3 is a marker for autophagosomes. And as you know very well, autophagy is a process by which cells eat themselves. They degrade intracellular organelles or particles in a regulated manner. So how could the LC3 levels be elevated in the myr-lysine knockout cells? Well, one possibility is that there is increased autophagy, and therefore there is more LC3. The other possibility, however, was that there is decreased fusion of autophagosomes with lysosomes in order to generate an autolysosome, which is where the contents of the autophagosome, including LC3, are degraded. So there was either more production of autophagosomes or less degradation. And we thought that this second possibility was more likely. And in order to analyze that, we used this probe, which is LC3 fused to two fluorescent proteins. A green fluorescent protein, which is sensitive to the acidic pH of lysosomes, and M-cherry, which is insensitive to the acidic pH of the lysosomes. So in the autophagosomes, you would see both fluorescent proteins, and therefore the organelles would look yellow, whereas once the autophagosomes have fused with the lysosomes to become an autolysosome, the green fluorescence will be quenched and you will only see red LC3. So we then analyzed the expression of this protein in wild type and myrlysine knockout cells, and we saw that whereas in wild type cells there were some yellow organelles, meaning autophagosomes, there were also some red organelles, meaning autolysosomes, after the autophagosomes fused with the lysosomes and the green fluorescence was quenched. Whereas in the myrlysine knockout cells, there were more fluorescent organelles, and most of them were yellow or green and red colocalized, meaning that there was a defect in the fusion of autophagosomes with lysosomes. So our interpretation of these experiments is that lysosomes move from the center of the cell toward the periphery by virtue of this complex that is regulated by Borg, and that this movement is required for fusion of the lysosomes with autophagosomes that form in the periphery of the cytoplasm, so that then the contents of the autophagosome, including LC3, can be degraded. If there is no Borg, the lysosomes remain clustered in the center of the cell, and these autophagosomes build up in the periphery of the cell. So in addition to autophagy, though, we found that there were defects in other functions dependent on lysosomes. We noticed when we stained the wild type and the myrlysine knockout cells for actin using phalloidin or for the cytosol using GFP, that the myrlysine knockout cells looked a little bit smaller. And I don't know whether you will be able to appreciate that in these images, but we quantified the footprint of these cells, the area on which they sat, by image analysis. And we found that indeed the myrlysine knockout cells were approximately 30 to 40%, they look 30 or 40% smaller than the wild type cells, as you can see here. However, when we measure, and this could be rescued by transfection of the myrlysine cDNA into the myrlysine knockout cells. However, when we measure the volume of these cells by flow cytofluorometry, by forward scattering, we found that the volume of the myrlysine knockout cells was similar to that of the wild type cells and the rescued cells. So what does it mean? That the cells have a smaller footprint but a similar volume. It means that they adhere less well to the substrate, that they are more rounded. And so they have a problem with adherence. Now, cell adhesion is important for cell migration. And therefore, we looked at the ability of the myrlysine knockout cells to move in comparison to the wild-type cells using various wound healing assays. And this is an example of one of these assays in which we plated cells around an agarose plug, and we grew them around this plug. And at time 0, we removed the agarose plug, and then we allowed the cells to fill the gap, to heal the wound, which happened over 30 hours for the wild-type cells. And as you can see here, the myrlysine knockout cells were less capable of healing that wound, consistent with a slower migration on this substrate. And again, this phenotype could also be rescued, at least partially, by transfection of the myrlysine cDNA. We also looked at these cells that were at the edge of migration by staining with antibodies to components of focal adhesions or components of the migration of the motility apparatus, such as paxillin. And we found that whereas these cells had well-organized focal adhesions, motile structures, as you can see here by staining with paxillin, in the myrlysine knockout cells, these focal adhesions were much less well-organized. And this could also be rescued by transfection of the myrlysine cDNA. And also, cell adhesion and migration are important for tumor growth and metastasis. And we therefore collaborated with Sylvia Gutkin, who at the time was at NIH and recently moved to UCSD, who used a mouse model of oral cancer in which HeLa cells are injected into the tongue of an immunosuppressed mouse, and wild-type cells will form a tumor over 10 weeks, as you can see here, whereas the myrtlysin knockout cells, although they grew normally in cell culture, they were unable to form a tumor in the tongue of these mice, perhaps consistent with their inability to adhere and to migrate. And also analysis of the lymph nodes in the neck show that whereas these cells, the wild-type cells form metastases, the myrtlysin knockout cells were unable to form metastases as well. So this Borg-dependent ability to move lysosomes to the periphery is also important for tumor progression and metastasis. So our interpretation then is that this Borg-dependent dispersal of lysosomes is important for the organization of focal adhesions, which in turn is important for cell adhesion and cell migration. And work done by others in the past had already suggested that late endosomes or lysosomes bring either adhesion molecules or signaling molecules to these focal adhesions, which allow for their dynamic reorganization in the process of cell motility. And finally, I want to show you another example in which, in addition to these functions, we found that the Borg-dependent dispersal of lysosomes is also important for lipid metabolism and transport. This comes from the observation that whereas wild-type HeLa cells have lipid droplets which can be stained with this dye, oil, red O. In red you can see the lipid droplets, and this is the nucleus. The myrtlysin knockout cells have fewer and smaller lipid droplets, and this phenotype can also be rescued by transfection of the myrtlysin cDNA. You can then extract these cells and then quantify the oil red by spectrophotometry. And you can see that there is like a 60 or 70% decrease in the amount of neutral lipids, which are the components, the main components of lipid droplets. So there is a defect in the storage of neutral lipids when you cannot move lysosomes to the periphery. We also quantify the levels of cholesterol esters, which is one of the neutral lipids that are stored in the lipid droplets, and we found that the amounts of cholesterol esters in the myrtlysin knockout cells were also dramatically reduced. And finally, we found another interesting cholesterol phenotype in these cells. We found that the amount of unsterified cholesterol found in late endosomes or lysosomes, as detected by staining with philippin, were increased in the myrtlysin knockout cells as compared to the wild-type cells, and this could also be partially rescued by the transfection of the cDNA for myrtlysin. So unsterified cholesterol in late endosomes and lysosomes was increased, free cholesterol was increased, and sterified cholesterol in lipid droplets was decreased, meaning there was a defect in the transfer of this cholesterol from the late endosomes and lysosomes to the lipid droplets. So we interpret these results to mean that the ability of cells to move lysosomes and late endosomes to the periphery is required so that these lysosomes can contact the cisternae of the endoplasmic reticulum, which are pretty much throughout the cytoplasm, so that there can be direct transport of cholesterol from the late endosomes to the endoplasmic reticulum, where cholesterol is again sterified by enzymes that are resident of the endoplasmic reticulum, and eventually stored in lipid droplets. If you are unable to move the lysosomes to the periphery, you will reduce these contacts, and you will also reduce the transfer of cholesterol from the lumen of the late endosomes and lysosomes to the ER and then to lipid droplets. So this Borg-dependent dispersal is also important for lipid metabolism, and we are also studying some of these other functions, but I think these serve as examples of functions that are very dependent on lysosome positioning and motility. So now, in conclusion, with words, we've then discovered this new complex named BORC, which is a multi-subunit complex that regulates lysosome positioning and motility. BORC mediates the recruitment of the small GTPase RL8 to the lysosomal membrane, initiating a chain of interactions that lead to kinase-independent movement of the lysosomes to the cell periphery. And this BORC-regulated lysosome dispersal is required for autophagy, cell adhesion, cell migration, tumor growth, and also lipid droplet biogenesis. And we also think that this process may be implicated in schizophrenia susceptibility because one of the genes encoding one of the subunits of BORC, the one that we call diaskedin, and that's also known as BORC7, is a major schizophrenia susceptibility gene in some genome-wide association studies. And for that reason, actually, there is a lot of interest in this protein. And finally, I want to thank the people in my lab who did this work. Most of this work was done by a single postdoctoral fellow, Jing Pu, and she had to work for a long time, actually, to figure out what was going on. From the finding of these novel proteins in the mass spec results to figuring out that there was a new complex and what the function of that complex was. And 3 other postdocs helped with some parts of this study. They are Rui Jia, Christina Schindler, and Jeannie Farias, and also one postbac student in my lab, Dylan Britt, helped with some of the experiments in neurons. I also want to thank my institute, NICHD, one of the institutes that is part of the NIH, for supporting this kind of basic research. And finally, I want to leave you with another message. Although we are now beginning to realize that lysosomes are highly motile, and this motility is important for many functions, there are people who have been thinking about this problem for quite some time. And among those people are this American rock band named Phish. Most of you probably don't know this band. The only reason I know them is because about 10 years ago I had children in college. And in college they were somewhat known. And already in 1994, Phish wrote in one of their songs, and this is not a joke, this is for real, they wrote, "They call him Lysosome, 'cause he runs so fast." So they already knew about lysosome motility. Runs like a junkyard dog with a brain of brass. And don't ask me to explain those verses because I don't know exactly what it means. But now I think I'm going to leave you a little bit with the music of Phish, and so that you remember what this talk was about. I don't know that I will be able to manage to do this. I cannot read.
They call him Lysosome because he runs so fast. Runs like a junkyard dog with a brain of brass. Go, go.
Thank you.
Really nice talk. So a couple related questions. When you look at the myr-lysin knockout or knockdown in axons, did you notice if there was any accumulation of synaptic components that might normally need to get turned over that might show up as morphological defects at synapses? And also, if you try to bypass this GEF idea with an RL8 GTPase defective, can it rescue the myr-lysin knockout?
Yeah, those are very good questions. We are now looking at the role of this whole pathway in regulating the motility of lysosomes in neurons. And we are sure that it plays a role, the whole pathway plays a role in moving lysosomes to the axon. Movement to the dendrites, though, appears to be regulated by a different process. Now, as to why are lysosomes going into the axon? So up until a few years ago, it was not known. Some people thought that if you need to degrade something in the axon terminal, you have to move those damaged organelles or something back to the soma for degradation, because that's where the majority of lysosomes are. But now it's becoming clear from our work and other people's work that lysosomes are able to enter the axon, and therefore must be able to encounter autophagosomes, for instance, or other organelles in the axon. I still cannot tell you exactly what the importance is for that movement of lysosomes into the axon, but we hypothesize that it will be important for fusion with autophagosomes, which have been shown to form in the distal axon, perhaps for growth of the growth cone during development of the axon, perhaps also for axon repair. And in fact, we are seeing that these, some of these lysosomes are fusing, in particular with the axon terminal. We see a lot of fusion taking place at the axon terminal. I cannot give you a definitive answer right now, but we have some ideas about what that could be. The dominant negative RL8 doesn't behave very well, and that's already known from work by Sean Monroe. It doesn't seem to be properly folded.
Okay, very nice talk, so thank you. So my question is related with the block 1 and the bulk 1. So as they share 3 common components. So this reminds me to a process, similar process from the early endosome to the late endosome. Because we know that the trafficking from the early endosome to the late endosome is through a maturation process.
Yes.
And this is mediated by also 2 very big complex, which also share some common components. So I'm not sure about the trafficking between the late endosome to the lysosome. So my question is, do you think the trafficking between the late endosome to the lysosome is through the direct fusion or maturation? So because I think if the maturation is true, so I think maybe some of the components from the bulk 1 may recruit some gap protein for the GTPase function in the lysosome. Yeah.
That's a great question that we discuss a lot in our lab. It's really, this doesn't really shed much light into the problem of late endosome maturation. So I can only give you my own opinion about it. I think that late endosomes, I think, both mature into lysosomes and can also fuse with pre-existing lysosomes. But that's my own opinion and bias based on everything that I've seen. And I recently had the opportunity to discuss this problem with people like Colin Hopkins or Lucio, who have been thinking about this problem for a long time. And this conversation convinced me that probably that idea is correct, that late endosomes both into a lysosome as their contents get degraded, but they can also fuse with other late endosomes. Now, I don't think that Borg is involved in that process, but perhaps the opposite movement, inward movement, is mediated by dynein, by RAB7 and RILP and ORP1L and other things that couple late endosomes to dynein. And probably those proteins will be important for the maturation of the late endosomes to the lysosomes. And one thing that I didn't show you, but that we do see, is a delay in the degradation of internalized EGF and EGF receptor. So there appears to be a defect in the delivery of EGF receptor and its ligand to lysosomes for degradation.
And I'm wondering how Borg is localized to lysosomes specifically. Is it lipid that makes it targeted, or is there another protein in the lysosomal membrane?
Yeah. We are thinking about all of those possibilities. First of all, the myristoylation in the amino terminus of myrlysine is important for membrane association and association with lysosomes. Second, there is another subunit, lyspersin, that also has the ability to specifically associate with lysosomes. And we think that that may be the key to the organelle specificity of this complex. And that subunit actually interacts with the regulator complex, which is involved in mTOR signaling. We are now investigating that association. I think that may also contribute to the recruitment of this complex to the lysosome. And as for association with lipids, we are still not sure. Block 1 is an elongated complex. It looks like a caterpillar, looks like a worm, and it's very flexible. Could possibly associate with membranes, and we've speculated that could be the case. And Borg, from what we've seen so far, also has a similar structure. But we have not seen any specific interactions with phosphoinositides or other.
When we constitute the migration along microtubules towards the plus end of lysosomes?
Yeah, it should be possible to do that. Up until now, yeah, some people are doing that actually. Yu Li in Beijing is reconstituting lysosome movement on microtubules, and I think that could be potentially very useful to dissect the biochemistry of the system. As you might imagine, there must be a lot of regulation going on here because lysosome motility is not just constitutive. It responds to starvation, it responds to formation of aggregates, and so there will probably be a lot of signaling going on the Borg complex or other components of this pathway, and that's something that would be very nice to dissect in an in vitro system. Christian.
So, beautiful story. So, on the Borg complex. So,
you know this story, yeah.
Yeah, nevertheless. So, it's always great to hear it then from the expert. So, the RL8 has been implicated to also then recruit the Hobbs complex, which is involved in fusion.
Right.
But I noticed that the lysosomal size looked like the same under these conditions. So what is your take then on this RL8 function? So I think it's clear that it has a migration role, but does it have a role beyond that as well?
Yeah, that is a great question. So the Hobbs complex, which is a tethering factor involved in late endosome fusion or fusion with lysosomes, has also been shown to interact with RL8. And we would think that if there is no Borg and therefore RL8 is cytosolic, there should be less fusion. The lysosomes look very similar. If anything, they have a little bit more material inside, and that may be the autophagy phenotype that we are seeing, but they don't look very abnormal. So I don't know what to say. We have not looked at the relationship of Borg and RL8 to Hobbs, so I cannot say much more about that. Yes.
I'm curious, is mitosis affected in myristin lockout cells? Because myristin, your data have shown that myristin lockout cells has reduced motility, reduced tumor size. Is it because of affected mitosis?
No, I mean, we haven't looked closely, but at least their rates of growth are similar. So we should look more closely, but we didn't see a defect in mitosis as far as we can tell. So also the cells, other than looking a little bit smaller, they are not multinucleated or don't seem grossly abnormal, other than for the phenotypes that I've shown. We think that perhaps another role for Borg and RL8 might be in the movement of synaptic vesicle precursors, because there is work done in C. elegans where they've shown that C. elegans mutants in RL8 and at least one of the subunits of what we now know is the Borg complex. At the time it was not known. Just a new protein. They have a defect in the movement of synaptic vesicle precursors toward the distal axon. So we think that may be another function of the Borg complex.
Thank you for your wonderful talk. And my question is about the phenotype of the complex. Because you showed that the single subunits knockdown showed a very, not that obvious, but very obvious phenotype relating with the lysosome positioning. So my question is that what happened to the single knockdown of subunits. The complex will not be together there, right?
Yeah,
Thank you.
the other subunits are degraded by the proteasome. When one is missing, the other subunits are unstable and they get degraded. We've done a lot of Western blot analysis for the subunits for which we have antibodies, and we see that when we knock down one, the others also disappeared, either partially or completely. So in the absence of one subunit, the others are unstable and they get degraded. That's why you see the same phenotype, regardless of what subunit you knock down or out.
Okay, any other questions? Well, if no more questions, let's thank Juan again for his excellent talk.
Thank you.